In this module, we are going to learn how a pest-resistant tobacco plant is created. Okay, a pest-resistant tobacco plant. This tobacco plant is infected by a nematode, and the name of the nematode is Meliugdanine incognitia. Meliugdanine incognitia. And this nematode, it infects the roots of the tobacco plant, which considerably decreases its yield. And this can be prevented by a technique or strategy called as RNA interference, or it is called as RNAi. It is called as RNAi. RNAi is a method of cellular defense. It's a method of cellular defense in all eukaryotes, which is taking place in all eukaryotes as a natural method of cellular defense. It prevents, what is RNA interference? It prevents the translation of a specific mRNA. We know once a transcribed mRNA is translated, only the proteins will be formed. Isn't it? Only after translation a protein is formed. So who gets translated? mRNA. So in RNA interference, it prevents the translation of the specific mRNA. It is silencing the mRNA so that it does not undergo translation due to the complementary double-stranded RNA molecule. We have learned in molecular basis, suppose RNA becomes double-stranded, translation won't take place. The same way in RNA interference, the specific mRNA is converted into a double-stranded RNA molecule by a complementary strand, and thus the translation of mRNA is stopped or it is silenced. The source of this complementary RNA, from where do we get this complementary RNA, is from the infection by RNA viruses or mobile genetic elements like transposomes that replicate via an RNA intermediate in our body. How do we get this RNA intermediate? That is by infection by an RNA virus or mobile genetic elements. They are called as transposomes, which are jumping genes which are present inside a cells. And these jumping genes or the transposomes or mobile genetic materials, they replicate via an RNA intermediate, and this is the source of the double-stranded, the complementary RNA which goes and binds to the specific mRNA and silences its translation. Using an vector, a gene of interest is introduced into the host plant, that is the tobacco plant. And what is used as a vector? What is used as a vector? We have learned that the vector for plants is nothing but Agrobacterium tumefaciens. So Agrobacterium tumefaciens, which goes and which is able to insert a gene of interest. And what is this gene of interest? It is the nematode specific genes. The nematode specific genes, that is a DNA, is introduced into the host plant using Agrobacterium. And this gene of interest which has been introduced into the host plant, it undergoes transcription to produce both sense and antisense RNA. Sense RNA and antisense RNA. And when the nematode comes and attacks this plant, in order to survive, it has to produce its own proteins. So the gene has to, a specific gene undergoes transcription to produce an mRNA. And only if this mRNA is translated, it will produce a protein which helps the nematode to survive as a pathogen in the tobacco plant. And the RNA which has been produced inside the plant by the introduction of a gene into the plant is complementary to the mRNA which is produced by the nematode. As a result, the mRNA of the nematode and mRNA which is produced inside the host plant, they bind to each other, they are complementary to each other, they form hydrogen bonds with each other and forms a double-stranded RNA. And thus, it initiates RNA interference, and thus the silencing of the specific mRNA of the nematode takes place. And what happens? The parasite cannot survive in the transgenic host. Who's the transgenic host over here? The tobacco plant, expressing specific interfering RNA. So that's tobacco plant is able to resist against the nematode Meliugdanine incognitia with the help of the technique called as RNA interference or RNAi. Thank you.